massive expansion and biobanking of stem cell-derived cardiomyocytes. A presentation from René Maas, PhD candidate, Department of Experimental Cardiology, UMC Utrecht. Despite the remarkable advances in the treatment of coronary artery disease, ischemic heart disease remains the most common cause of heart failure. Ischemic heart disease mostly appears as myocardial infarction, leading to the death of myocytes because of poor blood supply in ischemic conditions. Among patients with myocardial infarction, there is a strong relationship between degree of heart failure and mortality. The only therapy proven to reduce mortality for patients with cardiogenic shock is early revascularization myocardial infarction. Since this current intervention can't fully regenerate the infarcted myocardium, there is value in pursuing more innovative therapeutic approaches. Regenerative medicine is an innovative interdisciplinary method for rebuilding, replacing, or repairing the missed part of different organs in the body, as similar as possible to the primary structure. In recent years, regenerative medicine has been widely studied as a treatment for ischemic heart disease to repair the lost part of the heart by using stem cell-derived cardiomyocytes. Stem cell-derived cardiomyocytes can be utilized in various strategies, such as cell injections, 3D cellular patches or scaffolds, or paracrine stimulation. These novel, regenerative strategies could potentially lead to an improvement in the cardiac function of patients suffering from ischemic heart disease. The development of human-induced pluripotent stem cell-derived cardiomyocytes causes a breakthrough in the treatment of different cardiovascular diseases. Over the past decade, using developmental lessons, various protocols were established to ensure efficient differentiation of induced pluripotent stem cells into cardiomyocytes. Multiple embryonic pathways have been implicated in cardiac differentiation of pluripotent stem cells. Among them, Wnt signaling plays a particular crucial role during multiple distinct phases of cardiomyocyte development, and is necessary for cardiac specification, growth, and differentiation. Currently, the generation of stem cell-derived cardiomyocytes incorporates Wnt signaling activation from days 0 to 2, followed by Wnt inhibition at days 3 to 5, in order to induce a highly purified population of cardiomyocytes. A major limitation in the field, however, is the line-to-line variability in the efficiency of stem cell-derived cardiomyocytes generations, and the inability to effectively expand and passage-committed beating cardiomyocytes. To overcome this limitation, we developed an efficient method for the expansion of functional cardiomyocytes that are a reliable source for the downstream studies in a patient-specific manner. By using the knowledge obtained from previous studies in mice heart development, we found that concomitant Wnt signaling with low cell density culture resulted in a massive proliferative response of cardiomyocytes. Here, we use a cell seeding density of 2,500 cells per square centimeter and a small molecule to activate Wnt called CHIR. This proliferative response goes up to even 250-fold of cm number, produced from one cardiac differentiation. The expansion of functional cardiomyocytes can go up to a 250-fold increase of cardiomyocyte number within 3 to 5 weeks. In relation to costs, recent studies showed that transplanting 1 billion cardiomyocytes is not only challenging in the culturing but also costly. In the commercial field, rates for 1 million stem cell-derived cardiomyocytes costs range between $120 to $250, meaning at the in vitro and in vivo screening approaches will rise above the budgets of researchers. Using the expansion protocol, the costs of 1 million cardiomyocytes are now estimated at $2, which is approximately 100-fold less expensive than the commercial counterparts. However, for the regenerative applications, patient-specific and storage of cardiomyocytes is of great importance. Additionally, we improved the cryopreservation of cardiomyocytes, allowing efficient biobanking of cardiomyocytes. Now, cardiomyocytes can now very easily be enriched, stored, and expanded after thawing, with no need for immunosuppressing agents before transplantation, and fewer possibilities of genetic abnormality accompany them through multiple passages. Therefore, expanded cardiomyocytes are now suitable for downstream applications such as drug screening, sequencing, regenerative medicine, and 3D tissue engineering models. For the method described here, cardiomyocytes were derived from 3 different differentiation protocols. The B27, heparin and CDM3 based differentiation strategies were all suitable for expansion in cardiac expansion medium. Although the expansion capacity of the cardiomyocytes differentiated, using the B27 differentiation protocol appears to be the most robust. The cryopreservation steps allow for biobanking of large batches of cardiomyocytes that can be stored before expansion, or after passage 1 or 2. Thawing of cryopreserved cardiomyocytes is efficient. We obtain 74% viable cardiomyocytes. And during subsequent expansion, the cardiomyocytes exhibit a similar growth curve when compared to fresh unfrozen cardiomyocytes. Since the discovery of human-induced pluripotent stem cells, numerous strategies have been established to efficiently derive cardiomyocytes from these induced stem cells. In this video, we described a cost-effective strategy for the subsequent massive expansion of over 250-fold from high-purity cardiomyocytes. This effect is relying on two main aspects: the removal of cell-cell contacts and wind activation. This protocol maintains cardiac functionality, allows cryopreservation, and the cells can be used in downstream assays such as disease modeling, drug and toxicity screening, and cell therapy. You can read the full paper in Cell Star Protocols, March 2021.